Okay, so I guess we can start. So hello everyone and good afternoon. My name is Ronell Sicat from the KAUST Visualization Core Lab. So I'm a visualization scientist in this lab. And welcome to today's workshop on image segmentation, 3D analysis, and rendering. Yeah, so today, what are we going to talk about, right? So what am I going to show you? So the idea is hopefully you guys are working with some sort of image data, such as this. So image stacks, which is a bunch of 2D images. And you could be using some of the microscopes here, so electron microscopes, optical microscopes, or some of the CT scanners, right? So we have some micro CT, or other imaging techniques, right? So for your research, somehow you get some image data that looks like this. So this image stack captures 3D structures, right? And there's something in your images that you want to see, you want to measure, or you just have some questions about it, right? And our goal today is to figure out how to do those. So one of the things that you would typically have to do to start analyzing your data is a process called segmentation, right? So the idea of segmentation is just assigning each pixel in your image data to some label or material, right? So in this simple example, let's say of a scan of like sand particles. So you can assign your pixels to air material or sand material, right? Super simple. And what this segmentation or the label image allows you to do is then perform quantitative analysis on your data. So now you can more easily measure things, right? So you can measure the, let's say, the radius, diameter of your particles, the percentage, right? So what percentage of my whole region corresponds to sand and air, or maybe you want to count, right? Count how many particles there are to figure out the density, and so on, right? So that's the analysis part. So from your segmentation results, you can do some quantitative measures that give you your tables that you can then put in your paper, right? That hopefully answers your questions. And throughout this whole process, we have rendering or visualization, right? Which is basically generating images of your data, right? And the images hopefully show you what you're looking for in your data and helps you understand the whole workflow better. Okay, so this is kind of what we're going to do today. And we're going to look at different types of data, different ways to perform segmentation, right? Some are easier, some are more challenging. In the last example, we're going to use deep learning, which is an example of AI-assisted segmentation, which is like becoming more and more practical and popular these days. And we're going to look at different types of analysis, right? In different types of domain sciences. Okay, so this is what you can expect for today. The software, so the main software that we're going to use today is Avizo, particularly version 2024.2. So 2025.1 just came out, but we're still testing that. So, and it's not available in the IT website yet. So this one is available in the KAUST IT website. So this software is it's a commercial software, but fortunately for the people who are in the KAUST network, as long as you're within the KAUST network, you can use Avizo for free. So the software is sponsored and paid for by the KAUST Visualization Lab. So it's one of the services we provide. And it's a kind of complete application for image segmentation, analysis, and visualization. So you can do complete workflows using Avizo. Now, for those who are not in KAUST, there's alternatives, like free alternatives, like 3D Slicer, but we don't officially support that inside KAUST. But you can take a look at those if you want a free alternative. So before I get into the training, I just want to show some examples of what can you do in Avizo, what kind of visualizations, what kind of analysis, in what kind of domains. So for example, for the neuroscientists here in KAUST, if you have microscopy images of brain cells, for example, like this neuron, you can perform segmentation to extract and isolate the neuron, and you can perform different analysis, right? Like shoal analysis, which kind of measures the complexity of the branches. If you have rocks or similar type of materials, you can have CT scans of those and segment and analyze the porosity or the different components of your sample. You can work with corals, right? So a bunch of students CT scanned corals, performed the segmentation, and then it's not super obvious here, but given the segmentation, you can perform skeletonization, right? Which basically gives you the skeleton, right, of the coral. And this allows them to study how the coral grows. Okay? We're going to look at this particular example later. So this is a segmentation and quantification of mitochondria in mouse brain EM. And this is a particular example of a project that I worked on with a student here in KAUST a few years ago. So it helps you visualize the patterns, for example, in the materials that you create, and verify, for instance, that, okay, this is the pattern that we're expecting from the material, and measure some quantitative properties, right? So these are just some examples to give you an idea of what's possible, what kind of visualizations and analysis you can do in Avizo. So just quickly, I want to also introduce my lab. So I'm from the KAUST Visualization Core Lab, or KVL for short. So it's one of the 10 core labs in KAUST. And the core labs is a shared resource to all KAUST researchers. And we provide state-of-the-art facilities and expertise in different domains. So we have analytical chemistry, imaging, nanofab, plant growth, etc., supercomputing, and I'm particularly from the visualization lab. And there's 5 scientists in the lab, and we provide expertise in data visualization and analysis, but also data science, or AI, let's say, for more popular term. So we have our lead scientist who works mostly on visual analytics. So if you have or if you want to use dashboards, right, for understanding, let's say, tabular data, he's your guy. We have James, who's working on high-performance computing. So if you're doing computations and want to do visualizations on a cluster, let's say IBEX or our Shaheen supercomputer, he's your guy. And then there's me. I work with large data visualization. But I also do segmentation and 3D analysis, which is what the workshop is about today. But I also do AR/VR, so augmented reality and virtual reality. I'll touch more on that later. And then we have Didier. He's one of the AI guys, data science guys. He's more, let's say, his expertise is more on biological stuff. Let's say if you're interested in AlphaFold for protein folding, he's your guy. And finally, we have Gafoor, another AI expert. So he can help you with like image analysis using AI, or he's worked with genetic data and so on. So there's 5 of us with different specializations in visualization and data science. So feel free to reach out to us if you need help. In particular, our favorite thing to do is collaborate with researchers here in KAUST. So work on visualization or data analysis problems and publish papers together. together with KAUST researchers. Aside from the expertise, we also provide access to visualization facilities. So we have display walls. If you pass by building 1 across the spine, you're going to see these big screens, right? So these are high-resolution 2D screens that you can use for visualizing high-resolution data. And more recently, we introduced the AR/VR hub. So AR/VR for augmented and virtual reality. So we have headsets here. So we have Apple Vision Pro, MetaQuest headsets, HTC Vives. So if you want to do, or if you want to use AR/VR for your research, you can do so here. We have workstations. And these are not just for AR/VR, but also for Avizo. So for those who were asking earlier, if you don't have access to a workstation for using Avizo, you can book these workstations for doing Avizo. And particularly helpful are our segmentation tablets. So these are pen and tablet interfaces for doing manual segmentation. So I'll talk about those more later. So you can go to our website to book the facilities. And I'll provide the slides to you guys, so I'll post them in the website and send an email to all the participants so you can have access to this information. I just want to quickly show some examples just to promote some of the AR/VR data visualization services that we have. So if you're working with Avizo, it's actually super easy to look at your data in VR. Like matter of 2 minutes, 3 minutes, you're in VR with your data, like the example shown here. So this is a Avizo window, and I'm here with a VR headset interacting with my data and my project. Yeah? We can extract 3D models from your data, such as this 3D model here, and you can visualize it on a large semi-immersive cave display, which you can see here. It's great for presentations. We can do virtual reality or augmented reality applications that allow you to interact with your data in a different way, depending on your needs. So for example, here we have an example that allows a user to explore the internal patterns of fossils, right, using augmented reality. So if you have ideas for projects like these, you can approach us and then we can discuss possible collaboration. Finally, we provide trainings and workshops like this one. So I think this is the last one for this semester. So we have scientific visualization workshops, data science, and AI workshops. And almost all of our trainings are live, well, not live streamed, but recorded and archived in our YouTube channel, right? So you can take a look at that as well. So this is our website, and if you have questions or want to reach out to us, this is our email address. Okay. All right. Yes. So that's it for the introduction. So this is our schedule for today. So we're going to start with the basics of image processing, so understanding images. And basically, I'm going to show you the basics of Avizo because it's the main software that we're going to use today. So this is very important. After that, I'm going to show you 2 complete workflows. So two examples. One is segmenting and analyzing porosity in rock CT images. Who works with rocks here from ANPERC or petroleum engineering? Great. And we're going to look at mitochondria in brain EM images, electron microscopy. So who works with brains, mice brains? Great. So even though these are from like petroleum engineering and neuroscience, you're going to see that there's a lot of similarity, right, with different domains. So you're going to learn a lot from these 2 examples. All right, so let's get to the Avizo basics. Actually, before that, something very, very important. So these are very important concepts. I'm going to use a lot of the terminology here throughout the workshop. So I just want to make clear, right? So when you do your scanning on whatever device you're using, you get your image stack. Now, it could, I mean, it's very often that you get a bunch of images, right? It's a bunch of 2D images. And when you open them on your computer, you already see something, right? You see something. But under the hood, your data is actually just a bunch of numbers, right? I hope that's clear. It's a bunch of numbers. And in order for us to see something, right, to visualize it, we apply what's called a color map. So that basically applies a color to each intensity value in your data, right? So a very common color map is this grayscale color map, where 0 is assigned black, and your maximum value, let's say 255 or 100 or whatever, is assigned to white, and everything in between it goes from black to white, right? So that's why it's called grayscale. So you get something like this. So in this particular example, this is kind of a grayscale color map. It's possible, of course, to apply a different color map, like such as this rainbow color map. You probably see this a lot in a lot of papers. So the underlying data is still the same. You're just applying a different color map, so you visually you see something, right? And there could be a reason for that. So for instance, if you want to highlight or easily find like high intensity values, like red pops out. So maybe that's the reason. So yeah, and we'll see more of that later. And when you do segmentation, what's going on? So you want to create a new numerical representation of the data where you want to assign for each pixel a unique number, in this case an integer corresponding to a label, right? So in our example here, 1 is air and 2 is sand. If you have different data set, it could be like air and coral, or porosity and rock, or air, or I don't know, tissue and neuron, whatever. And yeah, once you do the segmentation, now it's easier to do your quantitative analysis, right? So for example, you can count how many pixels are in air, how many pixels in sand. You can measure fractions, you can measure distributions, etc. Okay? So again, image stack is your raw image. We do segmentation, you get your label images. And you do your analysis, and then you get your tables or whatever quantitative measures. Okay? Okay. I'm going to switch to my laptop here now. And yes, okay. So for this next part, I just want to give a quick demo, so you don't have to follow it. So actually, who has Avizo installed on their laptops now? Okay, so for now, for this one, don't try to follow. My goal here is to show you, to give you a good idea of a whole complete workflow, right? And a good idea of how Avizo works. So don't try to follow. You can follow in the next example, okay? And on the left here are just some of the modules that we're going to demonstrate in this example. Okay, so. All right. So once you have Avizo installed on your computer, you can have either a desktop shortcut or whatever, or you can search for Avizo. In this case, I'm going to use 2024.2. If you have other versions, it's going to show up here. But yeah, I'll pick 2024.2. And this is our kind of homepage. So can you read the text? I hope you can. But yeah, so here we have open data, open project, blank project. There's other things that I won't discuss here. I mean, you can get into them later on. But the first thing I want to do is open a data set. So I'm going to click Open Data. And I'm going to go to the datasets folder. Hopefully, for those of you who want to follow the examples later. You should have downloaded the data sets from the training website. If not, I'll provide the link again later for download. So in this case, I want to start with a sand example, and that's here. And as you can see here, the data set is in the form of multiple 2D images, right? So each file that you can see has a number that corresponds to the index in Z, okay? And I have some file here. Sometimes you're going to have like metadata files. So what you want to do here is just press Control A and maybe deselect the non-image data and click Open. And then Avizo will ask you about some properties of your data, like if you want to give it a name, what units do you want to use. So in this case, I'll stick with microns. And very important, if you're going to do some quantitative analysis, you have to set the voxel size correctly. So what is voxel size? This is the physical size of each pixel in your data. Pixel or voxel. Voxel is a pixel in 3D. So you should ideally know this, right? And it's usually something that's a property of your imaging technology, right? So your CT scanner or your microscope should give you this, okay? So let's say, I don't know, for example, I just use 2 microns. So this means each pixel in my data is 2 by 2 by 2 microns in physical size, okay? And click OK, and you get something like this. So yeah, so this is your main Avizo window. So let me just close this for now. Yeah, this is more like the default appearance of Avizo. And you can see here, this is called like the project view. So you can see it here. This is the project view where you have your data sets and different modules in Avizo. So this, when you load the data set, you actually get an icon like this that corresponds to that data set, right? So this icon represents your data. And when you click this icon, so when you click or select the data, you see some information displayed here in the properties window. So this is the properties window. When I click the data set, it shows me the size, right? So it's 128 by 128 by 128. This is the type of data. So it's a grayscale data, 16-bit. This is the memory. This is the physical size, right? So if you're going to measure this in the real world, this is the size, and this is the voxel size. It also gives you a preview of the histogram. Now, this view here on the right, that's our 3D view. And right now, you actually see a slice of the data, right? And I mean, I can move it in 3D. And that's coming from this module here. So actually, let me delete it first. So right now, I only have my data, and I don't see anything because there's no visualization module attached to it. So to create a visualization, you can click on this white, there's like a white circle on the right, click that. And then you get a list of modules that you can attach to it, right? So there's different modules that perform different things. So you don't have to know everything. I mean, I don't know everything, but for now, let's say I'll pick orthoslice. So this is what we saw earlier. So orthoslice, from the name, it's a slice of the data along XY, XZ, or YZ, right? So you can change the orientation. And because I click here, I see the properties of the orthoslice here, and I can change the properties, such as the slice number. So right now, I'm going through the data from top to bottom. So you can use this to inspect your data in 2D, okay? All right, and very important here is an important parameter is the color map. as I said, this is how you map colors to your data values. So for example, if I adjust the maximum value assigned to white, so it changes the contrast. And if I go to edit, it shows you the different color maps that you can use. So for example, I'll use the rainbow color map or physics color map. So you're going to see here a different mapping, right? And you can see, for instance, here, it easily shows you the high intensity values in red, yeah? But for now, I'll stick to the original, which is the grayscale. All right. So this is 2D, right? So this is looking at the data in 2D, but we know that the data is an image stack. It's 3D. So what we're going to do is I'm going to turn off the ortho slice. So you can use these blue like squares here to toggle the visibility. So I'll turn that off. Again, click on the data. Click here and look for volume rendering. So volume rendering is a 3D visualization algorithm. And it applies basically a color map, right, with the transparency. So let me just show you that. I'm not gonna explain the whole idea, but basically this is the color map. Yeah. So basically it's saying like values that are close to 0 will be dark gray and transparent. So this is the transparency. And everything close to the brightest value or highest value will be kind of bright white and opaque, right? And that's what you see here. That's what you get here. And you can see already, right? You get an impression of the structures in your data. So you can clearly see the sand particles, and maybe there's some noise over here, okay? Yeah, so if you have an image stack, so this ortho slice and volume rendering are, I would say, like one of the first modules that you want to use to investigate your data. Okay, so the next thing I want to do is now measure, right? I mean, what's my question about this data? Let's say I want to measure the fraction or like the percentage of sand. How much sand is there versus air in this data set, right? In this region. So what I have to do to answer that question is first segment the data, separate the air from the sand. And in order to do that, so let me turn off this first. So in order to do that, we're going to use our first segmentation tool. It's called interactive thresholding. Okay. So I'm going to, yeah. So interactive thresholding. So basically, it's just thresholding where it gives you control over the threshold values. So if I click on the interactive thresholding here, it shows me the values that I can change. And the most important part here is this, the intensity range, right? So this is basically the range of values that you are highlighting here. So you see here what's highlighted in blue. So if I move this, right, so it changes what I'm highlighting. And in this case, I want to highlight the sand, right? I want to separate it from the air. So you can manually adjust this to find a good threshold value. Or if I delete this again, I delete this and add it again, you can see that Avizo actually suggests some automatic threshold values. So let's say I'm happy with these threshold values and I click apply. So you're going to see that a new data set is created over here, right? So this is our original data. We apply thresholding. It creates a new data set. And this new data set is already my label image, right? So if I turn off interactive thresholding, now for this one, I can visualize it using voxelized rendering. So now, this is my label image. right? So it makes the, because I only segmented the sand particles, that's the only thing you see, and everything else is not visible. But what problem do you see here? Yes, exactly, right? So it's very noisy. So yes, you can do your analysis here, but you're gonna get some mistakes, right? Because you have this noise over here. So what we can do, so let me just turn this off. We can go back to our original data. So what we can do is apply filters, right? So the filters are basically algorithms or techniques that modify your image in different ways, right? So for instance, I can use a median filter. A median filter. So a median filter looks at a pixel neighborhood, right, and replaces the middle value with the median, right? And what this does is, I can show you the result. So this is the result. We can do a side-by-side comparison. Yeah, so on the left here is our original data. So I attached an orthoslice to it, and I only made that orthoslice visible on the left. Then we apply a median filter, and this is the result. Again, I attach another orthoslice to it and made it only visible on the right. And this allows us to compare the two, right? So this is very helpful when you're doing research, for example, and you want to have like side-by-side comparisons in your paper. You can do something like this. And okay, it's not super obvious on the projection, but you can kind of see that the noise is reduced. And that's actually more obvious if I, again, apply interactive threshold. Look at that, right? So there's no more outliers or kind of the noise outside. So if I apply, and now look at the result in 3D using voxelized rendering. So now you can see the result. It's very clean, right? So you don't have those outlier particles anymore. Okay, and this one, I'm more confident now in doing my analysis, right? And of course, sometimes you want to inspect your result in 2D. So this is 3D right now. So what you can do is turn this off. Turn off this. So I'll go to the orthoslice of the filtered data. And actually on the orthoslice, I can attach a color wash module. And for the color wash, I'll set the data to the segmentation. So what that does is it overlays the segmentation result onto your raw data. And you can blend them like this. So again, this is a nice way to show like segmentation results on your paper or on your supplementary videos. It might look a bit confusing, like I'm connecting too many things quickly, but hopefully you'll get used to that. And just remember that each icon here corresponds to a data set, an operation, or a visualization module. Okay? Yeah. So as I said, now let's say I want to inspect my segmentation result. So let me go back to a single window view. So I'm going to click this view and go here, single viewer. So I'm going to click the ortho slice for this and just go through the slices. Okay. So let's say for now I'm happy with this. So the next thing I want to do is do the analysis. So in this case, it's a very simple analysis. I just want to measure the fraction, like what's the ratio of sand to air in my data. So to do that, I click on my label image, this one, and I click here again on the white circle on the right, and I'm gonna attach a volume. Oh, it should be here, sorry, the labeled volume fraction. Yep. Okay. So volume fraction, and I'm just gonna run this, apply. Yeah, and might be a bit small, but here you get a value of 0.628. That means 62.8% of this volume is sand, and the rest is air. Okay. So this is a very simple but common type of analysis. And what else did I forget to say? Yeah, so we just did like a super simple segmentation and analysis workflow here, right? So starting from, just to quickly review, so we loaded our data. We used an orthoslice to investigate, to look at the data, right? And we used a volume rendering to inspect, to do an initial investigation of the data, like to make sure we see the structures that we expect to see. We used a thresholding module to do segmentation. I'll show you other modules and other techniques later for segmentation, but this is the easiest. We saw that the result was noisy, right, using voxelized rendering. So we said, okay, we're gonna clean the data first. So we used a median filter. And after applying a median filter, we saw that the data was cleaner. And then again, applied interactive thresholding on the cleaner data set. And finally, we got the clean segmentation that we're happy with. And using that clean segmentation, we did our analysis. In this case, a very simple volume fraction in 3D. And this gives us our final result, which is 62.8%. Okay. Yeah, that was a whole workflow for a super simple example in Avizo. And you might be wondering, right? Like, how did I know what modules to use? There's hundreds of modules in Avizo, right? So it's from, you can either read the user documentation, attend this workshop, and now you know the modules. It takes some time and experience to kind of know what modules to use. So for instance, in this workshop, I'm gonna show you, I don't know, 10 to 20 of the most commonly used modules. But in case you're not sure, you can always go to help over here. So if you click help, there's tutorials. There's a user's guide that's super useful. So if you click on user's guide, it brings you to this, this file, and you can search, right? So usually if you have keywords that are related to something you want to do, you can type here. So for example, let's just say segmentation, right? So it will point you to some segmentation tutorials, some more tutorials, and some modules, right? So there's a lot of resources here. Let's say you're interested in skeletonization. Yeah, so it has documentations here of the modules that you want to use. So hopefully you can come up with some keyword to search. But if you find that still difficult, search online. If all of that fails, then you can always reach out to us, right? And show us your data, tell us what you want to do, and we can guide you on what Avizo modules to use, and maybe like a workflow that would work for your data. So again, I want to emphasize that this is a simple example, right? The data it's kind of noisy, but it's super easy to clean. And it's meant to be simple, just to give you an overview of Avizo. So later on, we're going to look at more complex data sets. And let me just check my list of modules I want to show. So yeah, we have 7 more minutes before the next, or before the first break. So I want to show a few more things, right? So now I successfully separated my sand particles from my air, right? And I'm able to measure it. It's 62.8%. What if now I want to count how many particles there are? I mean, yes, you can do it manually, right? So I can count like 1, 2, 3, 4, 5, and so on. But what if you have like hundreds or thousands of particles? Okay, so that's the next thing that we're going to try to do. And for that, we're going to use a module. First, actually, let me show you. So the module is called label analysis. No, yeah. So we have to apply it to the label or the segmentation. So here, label analysis. Okay. And let me just click apply. All right. So the label analysis module will give you 2 outputs. One is a new label, and the other one is a table that you can see here on the right. And let me turn off this visualization and turn on visualization of this. So the label analysis actually counts or measures different things like volume, area, and so on for pieces that are separated in your data. So what you'll notice here is that this big chunk here, it's treated as one particle, which is bad. And these small pieces that are separated are treated as separate. So this is not a good result. So what we want to do first is separate the individual objects. And for that, there's actually an Avizo module. Guess the name. It's called separate objects. So I'm going to apply that to our segmentation data. And for now, I'll just keep the default parameters. You can play around with the parameters as needed. And now, let's visualize the result. It might not be obvious, but you're going to see the result here, for example. The module introduced this space between the particles. So let's look at the original again. So notice here, in the original, they're connected, also this. And in the separated, they're separated. So now, if we do label analysis on this one, apply, and visualize the result of that. Now we have something better, right? Okay, not necessarily perfect, right? Because this is still connected. Could be. But now it's more separated, right? And you can actually iterate on the parameters of the separate object to be more aggressive, to keep separating until you're happy with the results. But let's say for this one, let's say I'm happy for now, and you get a new table that's more correct, right? And it gives you the volume and the surface area of each piece. Now, what can you do with this table? You can actually export it. So if you click here, there's an export button on the top right. And you can export as CSV or Microsoft spreadsheet. I usually do CSV, it's just more flexible. And you can open that in whatever data, like spreadsheet analysis or plotting software you want to use, Excel, or you're using Python. The other thing you can do is to just quickly look at the distribution of values. So for example, I can click on the volume 3D here and click this plot a histogram button. And this plots the histogram like this. I mean, it's nothing super fancy now, but it gives you a quick overview of the distribution of volume, for example. Yes. And the final thing is, I mean, just to highlight, this thing that we did, this label analysis, is, I mean, from the name itself, it's another type of analysis, right, that you can do. And it's, I would say it's very typical. It's very common to use this, right, to quantify the different materials in your data set. So for example, if this is mitochondria or the pores in your rocks, you want to quantify those, like how big are they, what's the average, minimum, maximum, and so on. So those are all shown over here. The other thing I want to point out is you can actually customize the measurements you want to get. So if you click on label analysis, if you go here to measures, you can pick predefined measurement groups over here, or you can create your own over here. So you can come up with a list of things that you want to measure. So all of the things you can measure are here on the left. And you can, if you want to add something, you click on it and then you move it to your list. How does it work? I think here, add, I'll create a new measure so you can give it a name. I don't know, test. And then you can come up with an expression. So this is more advanced. Like if you have like a specialized type of measure, you can put like an expression there to compute the measure for you. Okay. And the last thing, we have a few seconds before the break. So let's say now you're happy with this result and you want to put it in your paper, right? So you want a nice image. Of course, you can always do a screenshot, like using the Windows snipping tool or whatever. But Avizo has a built-in like screenshot capture tool. So it's this icon over here, this camera icon. So if you click on that, you get a window like this. So you can specify the output file name and the format. You can put transparency. You can specify the resolution if you want. So if I do click OK, it should give me a file that looks like this that you can use in your paper. And it also works if you have multiple windows, right? So for instance, you want to show some comparison or like a 2D versus 3D. You can do camera and just make sure you check here, capture all viewers. And then OK. Yeah, and now you have your figure over here that you can use for your paper. You can also make videos. So if we have time later, I'm going to show you how to do that. But for now, let's take a 5-minute break, and then we'll go to the porosity in rock CT example. Okay? So I'm going to switch this to break. All right, so hopefully everyone is back. That was a short break, but we have to continue. All right. So hopefully those of you who want to follow along the hands-on today have already installed Avizo and downloaded the dataset. So you can use this QR code or go to the web page directly. So that's the training web page. So you have the links over there. And I will also post the slides in that page later. But for all those who registered, I'm going to send an email with the links. All right, so let's continue with the next example. So we're still going to work with rocks, particularly micro CT data. So this data set, I forgot to put the source here. It's from Domingo Latanzi Sanchez. So he's one of the micro CT guys in the petroleum engineering department. So if you want to CT scan rocks, he's your guy. So this is a data set from him. So it's a basalt rock sample. Okay. So I'm not a domain expert, but this is all I know. This is kind of a preview of what we want to do today. So in the simplest case, we want to segment and quantify the pores. So the pores are the air pockets, inside the rock. But more importantly, later on, we also want to quantify these regions. So hopefully you can see those. So between the air pockets and the rock, there's these parts here. They're like the in-between region where apparently you can imagine like some minerals growing, right? So it's growing out into the pore. And we want to quantify that as well, okay? So again, we want to quantify the pores and we want to quantify these in between. And the motivation behind this is we want to measure within this rock, how much gas can I trap inside, right? Let's say you want to trap, I don't know, carbon dioxide inside a rock. So you want to measure for every piece of rock a certain size, how much air pockets do we have? But since we have these regions, so these minerals growing, we also want to measure, because of these minerals, how much storage are we losing, right? But bottom line, we want to get to answer these questions, you just want to measure the percentage of the air pocket and these minerals with respect to the whole volume, okay? So that's what we're gonna do. Oh, that's for later. I'm gonna switch to Avizo. So we can just go to File, New Project, and I'm gonna discard. I'm not gonna save this project. And I'm gonna switch to single window. So here I'm starting with an empty project, and I'll just go click here, open data. And this time I'm gonna open the basalt file. And you're gonna notice here, this particular file has the Avizo icon already. And it's a .am file. So some of the scanners that we have here can already save the CT scan as an Avizo file. Probably a lot of the microscopes as well, because they're coming from the same company. So yeah, if it's in this same format, you're gonna see it like this, and you just click open, and boom, it's there. Again, Avizo automatically attaches an orthoslice. So you see something right away, and we can just inspect the data. So from top to bottom, it looks fine, right? And it looks very clean, actually, which is good. Sometimes you're going to see some artifacts. If it's noisy, you would know right away. So you can apply denoising algorithms to it. Or if there's some artifacts that you have to remove, yeah, you'll have to deal with those. So again, this is very typical. You inspect in 2D. And just to be sure, I would also inspect in 3D. So click the dataset and attach volume rendering. So looks fine. And again, what I want to quantify are the pores, so these dark regions, and these in-betweens, right? So the mineral. And basically, me personally, I usually always start with thresholding, right? So thresholding is just, again, the simplest type of segmentation tool. So I go with interactive thresholding, create. And here, you can see that it actually already suggests a range, right? In particular, it's giving us a range that segments both the rock, the basalt, and that boundary mineral, right? So I don't even have to change anything. I can just select this and apply. So now I have my result. I can turn off the interactive thresholding. And again, just to inspect the thresholding result, I go to my orthoslice. I attach a color wash to my orthoslice. And for the color wash, I can go to this drop down and say, use the thresholded data. Or instead of that drop down, you can click here on this white square here and click on data and attach it to your data source, right? So what this does is it overlays the grayscale with the segmentation, and if you click color wash, you can change the blending ratio. So for instance, let's make it 0.8. So my goal here is to just quickly inspect my segmentation. Is it good? Right, so particularly you want to focus on these boundary regions. So it looks really good. Again, what we segmented, so the label we have is for both the rock and this, I'll just call it mineral. I don't know what it is, but this mineral, we'll call it mineral, okay? So yeah, let's say we have that. And the next thing I want to do is, let's say I want to get the pores, okay? So what can I do? I have a bunch of options actually. So let me turn this off. So one option is, again, attaching interactive thresholding and actually flipping the range, right? So I'll copy this, paste it here, and then adjust the lower threshold. So it's a little bit small here, but you actually have a histogram of the data here to guide your range specification. But you can see, I hope it's obvious, I just changed the range, right? So it used to be like this, right? Like this big window. But I shifted it to the lower values. And now we're getting the pores, right? But, okay, so actually let's do that. Let's try this. Apply, and we get something. But now if I go to my orthoslice, So let's change the data. Instead of the one we segmented earlier, we'll go to the pores. So one thing that you'll see here, okay, maybe it's not super obvious here, but we actually have some, let's say, artifacts over here on the outer regions. That will be more evident if we do 3D. So look at that. So hopefully you can see and you believe me that there's kind of these artifacts outside, right? Why? Because, I mean, it's a boundary condition, right? Like the outside region can have low intensity values as well, right? And it can be interpreted by the thresholding as pores, right? And we don't necessarily want that. So a trick that we can do is, let's go back and take a look at the one we segmented earlier. So it looks like this. Now, if you look, again, let's go back in 2D. So in my head, I mean, just because of experience, I know that there's a module in Avizo that allows me to fill the holes of a label image. Okay? And if I can fill the holes of this one, and then later I can subtract that result from this, logically, I should get the pores, right? And that's what I'm going to do. So this is not magic, right? Like I came up with this because of experience. Like I know there's a module for filling holes, and if I do that, I can subtract. Now that you've seen it, and I mean you will see it, then you will also have that strategy with you, right? So later on, when you see a similar data set, you're gonna know, oh, okay, I've seen this before or something similar, and I can do that, right? So it takes experience. Anyway, let's take a look how that would work. So I would go to my label, and then I will attach the fill holes module. And you can fill holes in 2D or 3D, right? Like the interpretation of the algorithm can be done in 3D or 2D. Let's say I'm gonna do 3D. Okay, and now you get a result that looks like this, right? So it's the same as it was before, except the holes are filled. But I only want the pores, right? So what I'm gonna do is subtract these two, right? Or not really subtract, but extract the pixels where they are different. And to do that, we have an arithmetic module. Arithmetic. So I attach it to this one. And also, so that will be input A. Input A will be this, the filled. And then input B will be the one with the holes. And when you click the arithmetic module, it allows you to type in an expression that the module will compute. So in this case, I will just type A not equal to B. So for you, if you're wondering like, okay, what kind of expressions can I make? Click the help button. It will tell you about the arithmetic module. And here are the variables that you can use for the expression and the operations. Okay, there's quite a lot. This is super powerful, okay? Especially for the analysis side of things. So anyway, let's look at the result. So now if I look at the result in 2D, boom. I hope it's clear. So now we're left with the pores, right? And if you look at this in 3D, it doesn't have the artifacts of the boundary anymore, right? So again, this is just one, let's say, common strategy that you can use, right? So extract the, let's say, the rock or the enclosing material, right? If it's easy to extract, and then use fill holes and arithmetic to extract the inner smaller structures, right? So this is now our pores, right, inside the rock. But again, what will you notice here? You're gonna see that there's a little bit of noise, right? Maybe for your analysis, it's not critical to clean that up. But if it is critical, there's actually a module for that. So that is called remove small spots, right? So in many cases, the name of the module is quite intuitive. So if you search for it, you'll find it. But again, the more examples you see, the more workflows you see, the more familiar you will be with this module. Anyway, I click this remove small spots. And here there's a parameter of basically what's the limit of the, or what's the size of the small spots that you want to remove. Unfortunately, this is a bit more trial and error. So for instance, just visually here, I'm gonna say, okay, these are roughly, I don't know, maybe 50 pixels, like the small pieces I want to remove, and then I click apply. So you just have to try. You have to do trial and error, and then you get, because if you go too big, like some of the air pockets might disappear. Okay, so now we have a super nice and clean pore label, right? And again, just like with the super simple example we had before, you can already do the label analysis here. And yeah, I'll stick with the basic measurements. And then you get your table over here, which gives you your volume and area distributions. And again, you can visualize the different separated pores. Yep. And if this is the only thing you're interested in for your paper, take a screenshot or record a video. I'll show you how to do that later. And you're done. Another nice thing you can do in Avizo is you can actually create more interesting visualization. So for instance, I can blend my rock CT. So if I adjust the opacity here, I can blend I can blend the two. Oh, too much. Yeah. So you can create visualizations like this, right? So basically the grayscale volume rendering can provide context, right? So where are these pores situated with respect to the whole core, right? So you can do something like this and then take a screenshot or create a video. I mean, since we're here in the topic of visualization, another thing you might want to do is investigate inside, right? So you want to clip this volume and look inside. What you can do is attach an orthoslice, okay? And basically click this button here, clip. So that will clip the volume where the orthoslice is. And you can change the orientation. So that allows you to clip and look inside. And the nice thing is you can make this clipping plane invisible. So you only see a clipped version of the volume. Okay, so that allows you to investigate or take a look at your data from a different perspective. And if you're creating a video or animation for your supplementary material, you can also create an animation that does this. Okay? Okay. So far we just did the pores, right? But as I said earlier, we also want to measure that mineral. So this is more challenging, actually. Now I'll show you why. So let's go back to our original data. So this is our original data. I mean, before I continue, you'll probably notice, right? Like the more and more modules you have in the project window, it can get confusing. So, I mean, I have projects where there's like hundreds of these modules. You can like arrange them. There's a different view. I think this view, some people like this better. It's like a tree view. I like the other one better. But yeah, just you get used to it and just try to keep track. It also helps to rename the icons. So for example, this one I can click here and say rename, let's say original data, right? Whatever, like you can rename it to something that will help you understand or remember what's in there. Anyway, going back, attach an orthoslice. Again, we have the pores. Actually, let's go back to this. So we have the pores, and the next thing I want to do is segment this mineral in between. So let's try thresholding again, right? So this time, I mean, it's always worked for us, but this time I'll show you that this is actually very difficult. So let's zoom in. Okay, so I'll try to capture the mineral. So as you can see here, if you just use thresholding, right? So you can see that I'm capturing the minerals, which I want to get, but it's also capturing other things, right, that I don't want. And in fact, I mean, if I click apply and look at this in 3D, it's just a mess, right? It's a mess. So I don't want to use this. Now, another technique that I want to show you is applying filters to the images. Wait, yeah. So applying filters to the images so that the structures that you're interested in are easier to threshold. Okay? That might sound confusing, so let me show you. So the key to this technique is what's called a filter sandbox. So if you go to your original data and type filter, you can attach this filter sandbox. And when you do that, it's going to show this square, and you can actually move this square to a region where you want to apply a filter. Okay? So it's a sandbox. What does that mean? It means it allows you to experiment on different filters without applying the whole filter to the data yet. So if you click on filter sandbox, you can see the properties here. And here you're going to see filter, and right now it says none. So what you see here in the box is the original image. And let's say I'm just going to pick Gaussian. Okay? And I'm just going to exaggerate the parameters. Let's see. So I exaggerated the parameters just to show you that the filter is being applied inside this region only, and it's showing you how it would look like, right? But more important for me, because what's my goal? I want to see if the filter is helping me extract the mineral or not. So in the preview type here, instead of data, I'm going to say interactive thresholding. Okay? And I'm going to set the threshold to some value close to the mineral range. Okay? Oh, actually, this looks good, actually, but I didn't expect that. But anyway, but look at this, right? Basically, what this says is, hey, if you apply a Gaussian filter, which is basically, it's just blurring the data, right? It's just blurring the data. It's just doing some Gaussian weighted averaging. And if you apply thresholding with these values, this is the result you will get. And I mean, compare that with none. Let's go back to none. You see? It makes a difference, right? So let me go back to Gaussian. Actually, I was surprised by this. I didn't expect that, but it's still not good, right? Like, this is not good. So this is what I mean. So depending on your data, there might be a particular filter that would work best for you. And this filter sandbox allows you to quickly iterate over the different filters and different parameters. So you can take a look at the different filters and how they look like. Yeah. So for this one, I spent a few hours trying different things, and I found this particular setup gives me the best result. Okay, so it looks like this, and I have to modify the range. Yeah, it's not perfect, right? But for me, for now, it's good enough. And I mean, as you'll see in the next example, maybe you can use deep learning to even get better results, okay? But for now, I wanted to show you this. This is another strategy for segmentation without deep learning or machine learning, okay? So, okay, go back to filter sandbox, and let's say this looks good to me. All you have to do here is click apply. So once you click apply, the filter will be applied to the whole data set. And then you'll have to set the thresholding here. Okay, so yeah, looks good, right? I mean, more or less, you're getting kind of that mineral region. Okay, and for now, I'll click apply, and boom, I should have my region here. It looks good inside, right? Actually, that looks really beautiful. But you're gonna see we have these artifacts again in the boundary, right? Boundaries are most of the time challenging. But what can we do, right? So for this one, I'm gonna show you another tool, particularly for boundary regions. You can just crop it out, right? So you can just remove it from the label image. So for this one, you can use a module called volume edit. Okay, right? And you can actually specify a geometry, like a box. In this case, I want a cylinder, and I want it oriented along Z. So you can specify a geometry that you can use for cropping, right? I'm just going to change the projection to orthographic. And now I can adjust this. It's manual, so there's a bit of subjectivity involved, but that's fine for now. And I can be conservative, or it's up to you how conservative you want to be. And let's say, yeah, I'm happy with this one. I can just say cut outside. And this should give you now this, okay? So it's cleaner. I mean, there's a bit more here, so you can edit further. Let's do that. So it's up to you how much time you want to spend on refining your data, how critical. So this looks good to me. And go back to perspective. And boom, right? So now I have the mineral region. Yeah. Okay, so we're actually over the time here. So now I just want to quickly show how we do the actual analysis that we wanted, right? Because right now we have these two volumes. Let's Yep. So you can see here, I'm just disabling the different visualizations. Oops. Yeah, okay. So we have here on the left the pores that we did earlier. Actually, I'm gonna use this. Yeah. And on the right, we have the mineral stuff, right? The minerals. But I want to get the percentage of this and this in the whole data set. Now, if you remember from earlier, we had a module for doing this. What was it called? Volume fraction. Yeah. So I attach it to this one, the mineral. But the volume fraction, in order for it to measure this one, I have to combine them in the same label. But right now, I mean, under the hood, these pixels have a value of 1, and the background is 0. Same here, value of 1, background 0. If I naively combine them, it will mix up the two. So what I can do is use arithmetic, right? So this one is this. I'm going to attach arithmetic. And then input B is the mineral. So I'm going to have an expression that says A plus B times 2. Right? And what this gives me is something like, I'll show it on the left, like this. So the dark blue is actually 2. Let me do it like times 3 because 3 is red. Yeah. So the red is actually the mineral. And inside this, let's cut inside. Yeah. So inside it, you can see the blue, which is the pores, right? Does that make sense? So we just combined the two labels, the separate ones of the pore and the mineral, into one. And I assigned a value of 1 to the pore and a value of 3 to the mineral because, I mean, it's just for the color. Because if you look at the color map, 1 is like light blue, 2 is dark blue, and 3 is red. And you're going to see some greens. What do you think are the greens, which is corresponding to 4? So these are actually the pixels where they were both segmented as pores and minerals using our two techniques, right? And it's up to you to decide what to use in that case. So here I'll keep it and just say in the quantification, we'll say these are the uncertain pixels or voxels, right? So anyway, in the interest of time, I'm just going to do a volume fraction on this result. And if I just do apply over here, so sometimes it doesn't show the table here doesn't show the recent table, so you have to click show. So here is our result. There's actually something wrong here. and I will tell you what. So yeah, you will see here the labels. 1 is the pores, and it says 1.3%, and 3 is the mineral, and it says 2.7%, and then the 4, the uncertain. This is actually wrong because this volume fraction is measured with respect to the whole volume, including the empty regions. So what you want to do is in volume fraction, use, if you remember, the result of fill holes. Let me just show it to you on the left. Okay. We have this kind of label, which you can use as a mask for the volume fraction that says, hey, compute the volume fraction only with respect to this whole volume. That excludes the outside. And if you click apply, you should get the correct results. Okay? So this is the final correct result. So it's 2.1% pores and 4.2% mineral. Okay? And that's the answer to your question. Like, given, I mean, you'll have to divide it with some unit volume, but you can say for every unit volume of this rock, on average, we have 2.1% pores and 4.2% of the mineral. Okay? I hope that made sense. But yeah, this was the porosity analysis on the rocks. So far, we didn't use deep learning. So for the next example, we're going to use deep learning to try to perform the segmentation. So let's take a 5-minute break. And in the meantime, if there's any So again, I mean, if you have questions later on for your project, you can reach out to us using this email address. You can also check out our website. We have a highlights page there. So you're going to see some of the recent collaborations that we did. So you have an idea of what type of projects we work on. Okay. So let's go on to the next example because we have 20 minutes. So for this example, we're going to try to segment and analyze mitochondria in brain EM images. So the data set that I will work with here is from the Thermo Fisher, I mean, it's available in the Thermo Fisher website, but it's coming from a paper from some university, I can't remember, but they're cited in that website. So it's a publicly available data set. So I think I'm gonna skip this part because we only have 20 minutes. So this part, because the next tool that we're gonna look at is using the AI-assisted segmentation in Avizo. And this tool, it has its own user interface. So I wanted to give an introduction of that user interface, but since we're running out of time, I'll just go directly to it with the actual EM example. So just a quick overview. I mean, by now you guys for sure probably heard of some applications of AI, right? Artificial intelligence. So in our case, we're going to use deep learning models in particular for doing the segmentation for us, because sometimes thresholding, watershed, or the other traditional segmentation tools are just not enough, right? And what's the idea behind using deep learning for segmentation? So you have your raw data, so that's your image stack, and you have a deep learning model, right? So it's just a neural network, and in Avizo, we have some built-in models that are actually they're old-ish, but they're very generalist. So in my experience, they almost always work, okay? And the idea is you as a human domain expert will do a few manual segmentations. It's up to you how you do that. You can brush using the mouse or do whatever. And the idea is you just do a little bit of this, like investing maybe a few minutes and giving this to your model so that the model can learn how to segment the structures and then predict where predict a segmentation label or mask from what it learned from you, right? And this can be an iterative process, right? Sometimes maybe you don't have enough data, and then you get a poor result here, so you'll have to add more and more. Or you can also tweak the deep learning model to get better and better results. But this is the general idea. And Avizo has a simplified interface for doing that. And that's what I'll show you next. So that's called the AI-assisted segmentation. So let's get into it. So I'll go ahead and open our data set. So this one, we're going to use the hippocampus resampled. So I actually lowered the resolution a little bit from the original to make the data size more manageable. And this is how it looks like. And what we're interested in are the mitochondria. So basically these structures over here. Yeah. I'm not a neuroscientist. I'm not claiming to be. So I'm going to segment mitochondria based on like experience. I'm not saying it's super correct, but just to give you an idea, right? So these are mitochondria. So let's just go through the slice. Yeah, so it's a nice, super clean data set. And we can actually try, I mean, you know, since it's super simple, let's try thresholding, right? Let's just see what it's gonna give us. And here, you can kind of get a feeling that it's actually difficult, right? It's very difficult to extract just the mitochondria. So to me, it's clear that you can. It's not useful to do thresholding. Of course, you can try the technique I showed earlier, like apply a filter and try to maybe come up with a filter that will isolate the mitochondria. But for this one, we will try deep learning. So deep learning is really good at learning patterns, right? And for mitochondria, I think it makes sense because there's some sort of texture or pattern that is distinctive for mitochondria. And we will try that. How do you do that? So you just click your data set and then click segmentation. And this will bring you to this segmentation workroom, right? So actually, usually you're going to get a view like this. It looks overwhelming in the beginning, but once you get used to it, it's actually pretty simple. So you have your 3D view over here. You have different views of your image stack. Here is your XY, what's this, YZ and XZ. And for me, for now, I'll stick to the 2D, the XY. So I click that and single view. You can zoom in and out. You can change the slice number. And basically, on the top left here, you have your, let's say, parameters. So grayscale image, this is just which data set are you trying to segment. The label image, this is your like current output, which is empty right now. And then here in the labels tab, you have the list of materials or labels. So it says exterior and material 1. You can rename this to, let's say, mitochondria. Okay. So you can assign a color if you want. And numerically, these are represented as the exterior is 0 and mitochondria is 1. And if you add more materials, so if you create material, this will be 2 and so on. But for now, I just want 2 materials. So I will delete this, right click, delete. Okay. So right now it's empty. And in order to add something to my material, you can use the brush over here. So click brush. And now you see this circle. So that allows me to brush on my screen. So this red region here is a highlight. So of course this is wrong, so I will delete it, Control Z. You can set the size of your brush and zoom in. And I hope you can see it, but now I'm gonna start manually labeling my mitochondria. So you can encircle the boundary and click F to fill. So this is now the part where you have to invest some time and use your knowledge as the domain scientist to do your segmentation, right? So the labels that I'm generating now, I will feed this to my model to try to learn the pattern, right? So right now, I highlighted some regions. This is called a selection. It's actually not added to the mitochondria yet. You have to click this Add To button. So once you click this Add To button, it changes color, right, to whatever this is. Let's change it to green. So now when it's green, it means it's included as my mitochondria material, okay? So you can do more, but for now, let's say I want to use this to train my model. So how do you do that? So you actually can click here, this AI-assisted segmentation. There are other tools here that I will skip today because there's a lot. And the focus is this AI-assisted segmentation. So if you click that, you will get this window that says, what's the name of your model? So you click this button. So here, it actually gives you 2, 4, 5 options for the base models, okay? For now, I'll choose VGG19, but these are, I mean, if you look at the literature or if you're familiar with the literature, these are a bit old models, but they were quite useful, right? They are quite useful. So I'll click create, and it creates the model for me. So there's some parameters you can set, the number of training epochs. For the sake of time, I'll set this to 30. So this is the number of iterations, let's say, of learning for your deep learning model. So it's the number of times it feeds data into your model, adjust the weights of the model, adjust the weights based on the error or the loss. You can check this one to show a TensorBoard page. I'll show you what that means. And here, right now, we have one material. And click launch training. So it's warning me now that an ROI set is required. So what's an ROI set? I actually forgot that. So you have to click create new ROI set and create new ROI. This is actually very important. So this is the region. So you're telling Avizo, hey, I only have labels for this region, so please only use this part for training. So I'm going to pick this region. You can have multiple, but for now, I only use this. What you have to make sure of is, like in my example here, if this is part of my ROI, I should make sure that the mitochondria inside are labeled, because otherwise it will not. So this one I'm not sure, but let's leave it. Let's say like this, right? And now I label, and now I can go back to my training and say launch training. So this is the part that takes time, right? So if you have a computer with a good GPU, this will be faster. And the more data set or the more label data you have, usually the better your results, but the longer it takes for training. So this should take a few minutes. And it should show us at some point a TensorBoard window. For those who are familiar with like deep learning frameworks, this TensorBoard window will just show you kind of the progress of your deep learning training. Yeah, so it says training network started. Yeah, so it automatically opens this window where it will show you the current IOU score. This is intersection over union. It's basically how accurate the current model is. And it goes from 0 to 1. So right now, it's actually really bad. So what is this? The validation. OK. Training. It's really bad. It's less than 1. But I mean, less than 0.1. But it's going to evolve in time. It's actually ready. Okay, I mean, for some reason, it didn't show all the epochs. But it's done. I mean, the training is done with 30 epochs. And now we have a model. And we just want to see how good it is, right? So we're going to say, hey, use this trained model to create a new label and do it for all the images, so for the volume, so compute label. And this is now in prediction mode, right? So you trained, and now it's going to predict, and boom, look at that. This is surprisingly good. Can you see the result? That is incredible. And this was like a few minutes of training, right? Look at that. I hope, I mean, to me, this is like magic, right? But this is super good, right? I mean, of course, it makes mistakes, like here. And what can you do? You can further improve the model by going back here and adding more labels, right? So you can say ROI, create new ROI. For example, you can pick this region, right? Like maybe it didn't learn samples with this kind of background, right? So you'll have to label this correctly. Of course, that's wrong. But very important, right now, the active label is the result of the prediction. You have to go back to the original one, which is this, okay? And it should be empty here, okay? I mean, I'm not going to do it, but this is the idea. So you add this to the mitochondria label and then launch training. So it will retrain with the added data, and hopefully it will improve your results. But I will skip that now because we only have a few minutes. And once you're done, you go back to your project window, and now you have this hippocampus.ai assisted segmentation. That's your result. So if you look now in 3D, I mean, that's incredible. And this was like a few minutes, right? This is incredible. Of course, this is a clean data set. So it's an easy case, I would say, right? But it's not perfect, right? So there's going to be mistakes. And you probably want to proofread this. What that means is you can go back to this segmentation editor, load the result, and you can fix this, right, using the brush. So you can say, this is actually mitochondria, so you can add, or you can like say, oh, this is not mitochondria, so you can subtract. Okay, so you have to manually proofread it, right? There's also techniques that you can use. So for example, if you know that mitochondria has like a minimum size, you can use the remove small spots or remove islands module. So you can remove pieces that are like smaller than a certain threshold. There's also binary smoothing. So this one, you can, what this allows you to do, so I mean, you have to be careful about it, but it will remove pieces like these. These are like floating, because we expect the mitochondria to be like a 3D structure, right? So if you look at a voxel, it should have neighbors all around it most of the time. So what this binary smoothing will do is you can say, oh, let's look at 5 by 5 by 5 neighborhoods and only accept the label if more than, let's say, 30% of the pixels inside are labeled as mitochondria. So you see, it removes those like floating regions, but it also smooths the data, right? So it has certain limitations. But anyway, for the sake of time, let's say we're happy with this, or you did proofreading. All you have to do now to do your quantitative analysis is use you can use separate objects and then do label analysis. And then, yeah, you can get the distribution of, let's say, volume, right? So this is the distribution of volumes. You can get the number, right? Like how many mitochondria? We have 1,800 mitochondria in this cubic volume, whatever. So we're out of time, but that's it. Super simple. But again, this is a special example. I want to go back to my slides just to wrap it up. So again, as I said, I want to talk about this workflow recommendation. This is the typical workflow that I do if I'm dealing with a new data set. First, inspect the data. use orthoslice, volume rendering, check the histogram of the data, to get a feeling of what's in the data. Do I have noise? Is there something wrong with my data? If there's noise, you can use the filtering to denoise it or improve features, like highlight the features you were interested in. I mainly use the filter sandbox, but the common filters are median, bilateral, and non-local means filter. Check out the documentation to understand more what they are, or ask me. Depending on the situation, I would first try simple segmentation tools in Avizo, typically thresholding, like we've seen in the first two examples. Maybe watershed or texture classification. But these days, to be honest, most of the time I just go with the AI-assisted segmentation if the thresholding doesn't work, because it's super easy to use. If it doesn't work, try other AI segmentation tools, because we only have 5 models in Avizo. There's other more powerful models out there. I'll talk about some in a little bit. Worst case scenario, you don't want to do this, but I've had students who actually ended up doing manual segmentation. You have to invest days and weeks, but as you progress, you can try AI. The more data you have, keep trying AI. Not sure what to do? Reach out to us and we can try to figure out what to do for you. I just wanted to promote, if ever you need to do manual segmentation, sometimes the mouse is just not that great. So we have these segmentation tablets. You can book them using this link. They're in the same room as the workstations across the spine. Use those, right? There's another Avizo example available online, provided by Thermo Fisher. If you go to this link, and you have Avizo, you can download the example with the deep learning models and the videos and the PDF of how to do it step by step. So what I showed you today, the AI-assisted segmentation, it doesn't allow you to change many parameters of the model. But there's actually this DL training or deep learning training module in Avizo that allows you to modify a lot of the parameters. So check that out if you're interested. Again, link provided here. Other segmentation tools, as I said, 3D Slicer is a free open source alternative to Avizo, although we don't officially support it in KVL. I mean, we don't have much experience with it. Elastic, we kind of support this. So it's good for machine learning and deep learning based segmentation. Fiji, a lot of the, well, historically, the Imaging Core Lab was using this. MATLAB, we have a license in KAUST. There's a lot of available scripts and deep learning models now in MATLAB, and the others here. And yeah, that's it. Thank you for your attention. And let's collaborate. For those who are here, and if you have any challenging data sets, you can reach out to us to work on projects together. And check out our website to see our previous projects and to learn more about the lab. And reach out to us. Last favor, guys, if you have 1 minute, feedback form. It's a like 2-question thing, like 4-star or 5-star rating. And if you have comments, like, was it too fast, too simple? Like, this will help us improve the workshop for next time. Like, do you want to see examples from your domain or whatever? This is confidential, but it will help us a lot. So thank you very much for coming. I really appreciate it. And I'll be here in the next few minutes in case anyone have questions or want to chat. Thank you.